front of us is that it be resolved that animal models are an outdated paradigm for discovering cures and treatments for human diseases and testing drugs. And Dr. Greek will be the first speaker and will present his case. Dr. Greek.
Is this working? I think it's working. Is it working now? Test it. How is that? Can you hear me? Maybe push it up a little bit further. Testing 1, 2, 3, 4. Is that good? Is that good in the back?
Is the sound guy still here?
Is the sound guy still here?
I don't think it's on.
It's on.
It's on. There we go. Is that better? I don't know how to adjust it.
Probably on your collar.
Testing 1, 2, 3, 4. Is that better? I'll speak loudly. All right.
You could use the other microphone.
Be careful of feedback if you do this.
I tell you what, let's turn this one off.
No, it's on.
Yeah, but I was whizzing over.
Because your mouth is closer to the mic.
Okay, how's that? That good? All right. Okay, well, before I begin, I would like to thank the University of Buffalo for having me in the honors program, of course. I'd also like to mention the UB Spectrum for writing that very nice article. Sia Bond, the reporter, did a very nice job on that. I appreciate it. Of course, I'd also like to thank the Center for Inquiry and Andrew Skolnick for participating. David Triggle for being brave enough to be the moderator. Of course, I would not be here were it not for Dr. Nobis and Truly. They were very kind and allowed me to spend the night with them last night, and I do appreciate that. And finally, I'd like to thank you, the audience, for giving up your Sunday afternoon to come listen to a blowhard talk about scientific stuff. So, thank you. Now, the first slide, it says in summary what I'm going to try to explain in a very limited amount of time. And this is kind of sciencey stuff, and if you don't have a science background, I do apologize, but this is really the heart of the argument. And the slide says, because of differences in gene regulation and gene networks vis-a-vis evolutionary and molecular biology, we now understand why even two nearly identical complex systems, say a chimpanzee and a human, or even identical twins, may respond differently to the same stimuli, and hence why one complex system, or species, cannot reliably predict response for a different complex system, or species. Current biomedical research is studying disease and drug response at the level where the differences between complex systems, be they two different species or even two different humans, manifest. Hence, using animals as causal analogical models for human disease and drug testing is a scientifically invalid paradigm. And at the end of the lecture, we're going to see that same slide again, and hopefully you'll know what all those little words mean by then. Our second slide, before we talk about using animals as causal analogical models, I need to define our subject a little bit better. Animals can be used in roughly 9 different ways in the pursuit of scientific knowledge. Now, what I am going to be addressing today is only numbers 1 and 2, and you might want to refer back to this slide on your handout as I continue the lecture. I am going to be addressing the use of animals as models for human disease and animals as test subjects or models for drug testing. And I'm going to claim that is scientifically invalid. Now, that leaves us 7 ways that animal rights people might object to, to animals being used, but these 7 ways are scientifically valid. For example, you can use animals as spare parts. I'm from California. Our beloved governor recently had an aortic valve replacement, and where did that valve come from? A pig. Yes, I've heard all the jokes. Animals can be used as factories. For example, animals were used for decades as a source of production for insulin, and many lives were saved because of cow and pig insulin. Animal tissue can be used to study basic physiological principles. Animals can be used for dissection. They can be used as a heuristic, and so on and so on. All of these, numbers 3 through 9, are scientifically valid, okay? But they are not what we call causal analogical models. That's what CAMS means. And it will be my contention that when animals are used as CAMs, that is scientifically inappropriate. Okay, there's 5 parts to the lecture, and again, I apologize because I've got to go pretty fast. There's 5 parts to the lecture, and the first part is that physics sets the standard for science. Sir Isaac Newton said many years ago, he said, quote, "Therefore, to the same natural effects, we must as far as possible assign the same causes." And he went on to say that this rule applies to respiration in man and beast and the descent of stones in Europe and America. Now, Newton and his follower, Claude Bernard, who really made animal experimentation what it is today, he's really the father of modern-day animal experimentation, believed in what is called the resolutio compositive method of study, or what we might today call the method of analysis and synthesis. And this revolves around causal determinism. Again, if something caused X to happen in Europe, that same thing would cause X to happen in the US. Well, Newtonian physics is and was a wonderful paradigm. But other paradigms have come along since Newton, including relativity, quantum theory, chaos theory, complexity theory, which we're going to discuss a little bit, and string theory. Now, none of these things have replaced Newtonian physics, but they have expanded on the way we look at the physical world. So, let's talk a little bit about what complexity is. Today, you see a lot in the press, in the literature, both scientific and lay literature, about complex systems or complexity. And these are some of the characteristics of a complex system. Emergence is one. Of interest to us, perhaps, is the fact that relationships are not linear in complex systems. A very small stimulus, for example, may cause a very large effect, or it may cause no effect at all. And extrapolating from one complex system to even an almost identical complex system is very difficult. The relationships contain feedback loops, and most importantly, vis-a-vis reductionism and determinism, the parts cannot contain the whole. Newtonian physics was all about reductionism and determinism, and that was what Claude Bernard took from Newton and applied to research with animals. And it worked very well for a while, but as we will see as you study complex systems, like this one, the paradigm fails. Now, the causes, this is a complex system. This is actually a gene in a fruit fly. And you can see there's a lot of lines that mean that these genes interact with each other. There's feedback loops. Sometimes you can delete one of the genes and have no effect at all. Sometimes you can delete the gene and you see a huge effect. This is a diagram of a complex network. The causes and effects of the events that a complex system experiences are not proportional to each other. The different parts of a complex system are linked to and affect one another in a synergistic manner. There is positive and negative feedback in a complex system. The level of complexity depends on the character of the system, its environment, and the nature of the interactions between the system and the environment. And again, this leads us back to nonlinearity. Now, in the past, society has learned to understand reality through simplification and analysis vis-a-vis reductionism, and it worked great. Some important simple systems are successful idealizations of primitive models of particular real situations. For example, a perfect sphere rolling down an absolutely smooth slope in a vacuum. Now, this is the world of Newtonian mechanics, and it ignores a huge number of other simultaneously acting factors. Although it might sometimes not matter that details, such as the motions of billions of atoms dancing inside the sphere's material, are ignored, in other cases, reductionism may lead to incorrect conclusions. Now, this is especially true in complex systems. In complex systems, we accept that processes that occur simultaneously on different scales or levels are important, and the intricate behavior of the whole system depends on its units in a non-trivial way. Here, the description of the entire system's behavior requires a qualitatively new theory, because the laws that describe its behavior are qualitatively different from those that govern its individual units. What we are witnessing in this context is a change of paradigm in our attempt to understand the world. The laws of the whole cannot be deduced by digging deeper into the details. And there's a name for this. It's called systems biology. And Leroy Hood and others are founders of this field. So, as with chaos theory, the behavior of a self-organizing complex system cannot be predicted by studying a different system. And complex systems do not observe the principles of additivity. And what I mean by that is their components cannot be divided up and studied in isolation vis-a-vis reductionism. As I said, the causes and effects of the events that a complex system experiences are not proportional to each other. The different parts of a complex system are linked and affect each other in a synergistic manner. And I hope you can kind of understand what I said when you look at that system of genes and how they interact. If you can't, I brought another slide that looks remarkably like the first one. Okay. Now, living systems, such as chimpanzees, mice, and humans, are obviously examples of complex systems. It should be equally obvious, therefore, why extrapolation between species will be problematic. Small changes on the genetic level can lead to very large differences between species. Indeed, that's what evolution is. So the claim that humans and rats are the same animal, just dressed up differently, simply isn't true. Moreover, it is irrelevant to point to observed similarities in genetic makeup between species, since the details of the differences are in the interactions between the conserved genes, not in the genes themselves. It is as though humans and rats have a common genetic keyboard on which different phenotypic tunes are being played. What matters is not the similarity with respect to the keyboards, but the difference with respect to the order and timing of the activation of the keys. Predicting human response based on an animal model is not an example of applying a relatively simple set of well-established scientific principles as we see with Newtonian physics. Indeed, living organisms are much better examples of complexity theory than Newtonian physics. Using the example of a model airplane, as so many defenders of the animal model do, is a good example of this. Studying a model airplane, especially a paper airplane, will allow the observer to demonstrate the basics of flight. Bernoulli's equations hold true. But if anyone seriously believes this method can be or is used to build or repair a 747, they're deluded. Just as animal models can be, and indeed were used to demonstrate very basic facts concerning anatomy and physiology, so a model plane can be used to demonstrate basic physics laws concerning flight. But today, when we want to know why a 747 crashed, we don't build paper airplanes, and neither should we suggest to the public, who is paying for animal experiments, that a cure for AIDS, cancer, or stroke will be derived from animal models. Now this leads us to part 3, which is the regulation and expression of genes. The mouse, we know, has about 30,000 genes. We also know that humans have about 30,000 genes, and what's more, 99% of those genes are shared. And what I mean by that is humans have about 300 genes that mice don't have, and mice have about 300 genes that humans don't have. Mice and humans both have the same gene that when activated in mice grows a tail, okay? in humans, perhaps with the exception of myself, don't grow a tail. I've been accused of having horns and a tail. But most humans do not grow a tail even though they have this gene. And the reason they don't is because the gene is not turned on. Now, in the old days, if you look to the left of the diagram, this is how we thought biology worked. A single input, whether it was the environment or a genetic switch or a genetic signal, led to a single output. Today, because of the Human Genome Project and other research, we know that genetic networks actually control what goes on in the body. And these genetic networks have multiple inputs and multiple outputs. And even if you have a genetic network that is identical in a mouse and in a human, because of all this interaction that's involved, you can get two entirely different outcomes from the same stimulus. Not only that, but because of very small differences in just one gene, say the gene at the top, you can have a cascade effect that will turn the stimulus into something very different when it gets to the bottom. So genes work in networks, they're interconnected, one influences another, and any small change at the appropriate time will result in a cascade effect that will have major implications. Now this is kind of a little mathematical formula of what I've just said. If you have inputs or stimuli or genes, A sub 1 through A sub n, that are interacted on by environmental factors or other genes for that matter, as represented by B, you can have multiple different outcomes, even though you start with the same conditions. Well, here's what that means. This is a slide that was in Drug Discovery Today. It's a magazine that relates to drug development. And this was a slide that was done by Dr. Bob Coleman in his article. And what he looked at was one gene, it's the CFTR gene, and he assayed the amount of this gene that was turned on in mice and in humans in all these different tissues. Now, as you can see, in every single tissue, the gene is present. But in the mouse, except for the salivary glands, which is the first one, in the mouse it's turned on far greater, sometimes by 10,000-fold, than it is in the human. Okay? This is the difference between you and a mouse. Okay? Same gene, regulated and expressed differently. Now, the Public Library of Science has a new journal called Biology, and in an editorial, they said this about mouse models' autoimmune diseases. And they were basically talking about all the things that I've talked about so far. They said, these results fall in line with mounting evidence that background genes are not silent partners in gene-targeted disease models, but can themselves facilitate expression of the disease. This finding underscores the notion that genes are not solitary, static entities. Their expression often depends on context, with genetically complex diseases having the requisite combination of susceptible genes does not always lead to the disease. And again, we get back to the network. Same network in you, same network in me. I may suffer from cancer, you may not. Okay, part 4. How are animals used in biomedical research? The way that they are primarily used is as causal analogical models. Causal analogical models assume several things. Number 1, the first condition that must be met in order for a thing to be considered a CAM is that X, the model, is similar to Y, the object being modeled, in respects A, B, C, D, and E. Now, we know that X, the model, also has additional property F. So, while F has not been observed directly in Y, we reason causally by analogy that Y also has property F. And I'll give you one just simple example. Guinea pigs and humans have a lot in common. So if you give a drug to a guinea pig and it kills the guinea pig, you might want to think twice about giving it to the human, right? Make sense? Unfortunately, penicillin kills guinea pigs. And that's what's wrong with the theory. Animal models suffer from what is called causal functional asymmetry. What we assume when we do an experiment on an animal is that the same function in species has the same causal mechanism. But we know this is not always the case. For example, causal functional asymmetry notes that the same function can be derived by different evolutionary pathways. For example, birds ventilate through an entirely different causal mechanism evolutionarily than humans, but they accomplish the same purpose. And there are many other examples of this. So causal functional asymmetry has major implications for extrapolating data between species. Now, CAMs must satisfy two further conditions. The common properties that the model and thing being modeled share, A through E, must be causal properties. You can't just say the guinea pig has a heart, humans have a heart. The properties that you're comparing have to be causally related to what you're studying, what outcome you're considering. And number two, the properties, the common properties must be causal properties, which, number two, are causally connected with the property F that we wish to project. So specifically, F should stand as the cause or effect of A through E in the model. And this leads us to systemic disanalogies. Complex systems, like rats and humans and mice, are by definition going to be different in very subtle and unknown ways. We do not know everything there is to know about Drosophila. We certainly don't know everything there is to know about Homo sapiens. And we know for a fact that the same exposure often causes very different reactions in different species. So for a CAM, or an animal model, to be predictive, there should be no causally relevant disanalogies between the systems. And this is just flat out impossible until you know everything there is to know about each system. You can usually find an animal that will mimic a human response. Mice respond pretty much the same way to penicillin that humans do. The problem is you can't have that knowledge except in retrospect. Okay, part 5. This is the last part. So far, the discussion has been somewhat abstract. I'm sure I've explained complexity theory to you such that you now can all go home with a vast understanding of it, but perhaps my skills are not quite that good. But this slide hopefully illustrates in a more tangible way the problems of complexity and hence of extrapolation. Of 10 medications that were withdrawn from the US market between 1998 and 2001, 8 were withdrawn because of side effects that occurred almost exclusively in women. Okay? Men could take the drug and by and large do well. Women took the drug and by and large died or wished they had. Now, men and women are obviously similar in terms of evolutionary biology and gene regulation, but they responded very differently to these drugs. Similarly, a study in Science revealed that one strain of mouse could have a gene removed without obvious adverse effects, while another strain would die without that same gene. Now, a strain of mouse is like a breed of dog. Okay, that's basically what you're talking about. The anti-cancer medication, Iressa, was thought to be ineffective, but upon further analysis, it was found to be very effective for people with a specific genetic mutation. Now, today, because of pharmacogenetics and other advances, we are on the verge of what is called personalized medicine. And what this means is that drugs and treatments can be tailor-made for you as an individual. This, again, illustrates how very small differences between complex systems, in this case humans, can result in profound differences in disease and drug response. If men cannot predict the effects of a drug for women, and one strain of mouse cannot predict what will happen to another if a gene is removed, perhaps we are studying organisms at the level of organizational complexity that defines not only one species from another, but actually defines one individual from another. Evolution, complexity theory, and molecular biology predict that animal testing should not be an effective means of conducting biomedical research. And most importantly, empirical data supports this. Most physicians will tell you that they do not even look at the results from animal-based experiments because it does them no good in their clinical practice of medicine. Now, since the early days of Newtonian physics, important implications for causal determinism theory in biological science have come to light. And this has mainly happened because we've united Darwinian evolution with our knowledge of DNA, genetics, genomics, evo-devo, complexity theory, and so forth. First of all, we know that animals are examples of complex systems, and that therefore small differences between them can lead to changes in the system as a whole in a non-linear fashion. An example of this would be the fact that most of the time, identical twins do not suffer from the same severe diseases, such as cancer or multiple sclerosis, and obviously only a very small difference accounts for this, because we're talking about identical twins. Gene expression has been studied in numerous animals, and the same genes found to express very differently depending on the species, as we saw in that slide. Again, this is because very small changes between initial states in complex systems can propagate into extreme variations. Number two, the level of examination has changed since the 19th and early 20th century. Our examination of living systems has become increasingly fine-grained. We have found that subtle differences between organisms tend to outweigh gross similarities as explanations for biological activity. Science could and did use animals to shed light on shared functions, such as the basic function of the liver or pancreas and so forth. But today we're studying drug response and disease at the level that defines not only a species, but most of the time actually defines the individual. Now, the intact systems argument has historically been the animal modeler's main argument, and it goes something like this. We must test on animals because no experimental system, be it in vitro, in silico, mathematical modeling, and so forth, can predict what a drug will do to the intact living human system. That's basically the argument. Now, ironically, it's the fact that each intact living system is a different complex system that invalidates the use of animal models. Complex systems, as we have seen, are more than the sum of their parts, and different complex systems respond differently to the same stimuli. The implicit claim in the intact systems argument that humans and other animals are the same biochemical animal just dressed up differently simply is not true. Yes, we all have mitochondria, we have ATP, we're made of cells, and so on and so forth, but that is not the question that we're addressing today. Today we're addressing the question of drugs and disease. When we humans were ignorant of the function of the lung, this reductionistic approach vis-a-vis Newtonian physics worked very well. But the level of our examination has so greatly advanced that interspecies extrapolation is no longer useful. And as I said, it is irrelevant to point to observed similarities in genetic makeup between species, since the reasons for the differences are in the regulation of these conserved genes and the resultant interaction between them, not in the genes themselves. As I said, it is as though all species have a common genetic keyboard on which different phenotypic concertos are being played. What matters is not the similarity with respect to the keyboard, but the differences with respect to the order and timing of the pressing of the keys. Studying Mozart's keyboard could not have predicted Ray Charles. The piano itself does not predict the noise or music that emanates from it. Though genes are causal, as the PLOS Biology Journal said, they alone do not determine outcome. Early on, medical research was almost solely the domain of the experimental physiologist. But today, medical research, even the theory supporting it, is multidisciplinary. Physicists and mathematicians are involved vis-a-vis complex system analysis. Evolutionary biologists, molecular biologists, mathematicians, computer scientists, physicists, and many, many others all play different but vital roles. It is incumbent upon all involved to understand the implications of knowledge gained from other fields. And that means that researchers should abandon modes of inquiry based on unexamined assumptions from the 16th, 17th, and 18th centuries. Historically, medicine has been practiced based on statistics. If you suffer from high blood pressure, and research showed 98% of people with high blood pressure responded to medication X, then you wanted medication X. Now, you might be among the very small minority that responded better to medication Q. But because we had no way to determine that, I would prescribe for you medication X. Today we are on the verge, indeed we are experiencing personalized medicine. That is drugs designed, tailor-made for you. Not me, not your mother, not your sister. So, in light of that, considering the implications of personalized medicine, basing treatments on the response of a different species is like using phrenology to study mental illness or trephination to cure malaria. It simply isn't going to be effective. So, finally we come to the harm to humans from studying animals. Now, one question I'm often asked is, why don't we do animal experiments? So what if we waste a little money? Big deal. Well, I grant you, as an American taxpayer, I'm accustomed to the government wasting my money. But when we experiment on animals and we extrapolate those results to humans, humans die. And I have a problem with that. And these are just some of the examples. Smoking was thought to be non-carcinogenic for years because we cannot reproduce lung cancer in dogs. High cholesterol diets were recommended. Indeed, we were giving high cholesterol, high fat diets to patients who had just suffered heart attacks. Asbestos and many other environmental induced cancers were given to the general public without any hesitation because of studies done in animals. And again, we can go on and on and on down the list. To put it in a little bit more modern context, this is a study from Science in 1997. And the National Cancer Institute tested 12 drugs that were currently being used successfully to treat cancer in humans. And they studied mice that were called nude mice or xenograft mice. And these mice were growing the same cancers that these drugs were used to treat. And indeed, they were human cancers because they took a bit of the human cancer, implanted it into the mouse would grow it, and then the scientists would give the mouse the appropriate drug. Well, what this study showed was that 30 out of 48 times, the drugs were ineffective. Okay? So 63% of the time, even though we knew that these drugs were effective in treating human cancer, they were ineffective in treating mouse cancer. Now, this same article goes on to suggest that perhaps we have lost cures for cancer because they were ineffective in mice. Okay, that's the National Cancer Institute saying that. That's not me. Drug testing has come under fire for many decades. Here's just 4 studies, and there's about 10 to 20 studies out there that have looked at drug testing just like these have. I picked these because they really show the range. These studies show that animal models failed to predict drug toxicity between 43 and 95% of the time. And you'll see they failed to predict 43%, 67%, 83%, 95%. I don't care how you do the statistics, it doesn't work. Okay, predicting toxicity based on animal models is a failed paradigm. So why should we abandon it? Well, very simply because it doesn't work. Okay, I don't care what alternatives you have. We don't do bloodletting today. We don't do phlebotomy to treat malaria. And the reason we don't do that is because it just doesn't work. All right? So regardless of all the other reasons, animal experimentation using animals as cams to study human disease and test drugs should be abandoned. It diverts money from viable research modalities. We have better options. It misleads researchers, and so on and so on and so on. Okay. Now, does this slide make a little bit more sense since you saw it for the first time? I'm running out of time, so I'm going to go ahead and go to the next slide. This is a very complicated subject, okay? I essentially have read hundreds of books to get the knowledge that I need to give even this very short, much too fast presentation. And if you really want to understand why the animal model is not a scientifically valid paradigm, you have to know about gene networks, you have to know about gene regulation, you have to know about complexity theory, you have to know about Evo-Devo, and these are just some of the books that we recommend that you read, and we have a much longer list on our website. Let me close with this slide. What a difference a little DNA makes. Thank you very much.
Thank you, Dr. Reek. I will now call on Andrew Skolnick, but it will take a couple of minutes. I think he needs to set up his PowerPoint.
I think it's set up.
Set up, okay?
You know, maybe a couple of minutes.
Before I start the clock, have a couple of minutes.
Okay, so, hold on a second. There's a touchpad in the console that you switch from one computer to the other. Oh, hey, this is where, there we go. Was I introduced?
I did introduce you, and I said you have a couple of minutes while you set up your PowerPoint. You have,
Paying attention.
you now have 40 minutes.
Thank you. It's a honor to be here, taking part in this debate. I want to thank Dr. Nathan Novus and Dr. Ray Reed and others who made this possible. Is there an echo? I'm getting or am I misspelled here? I am a medical journalist who for more than 9 years served as an associate editor at the Journal of the American Medical Association, which gave me a unique position as a journalist, an investigative reporter to observe medicine at its best and at its worst. I understand a lot of what's wrong with the American medical system and also with medical research. But I don't agree with Dr. Greek's arguments that because the water is a little murky, we need to throw out the baby with the bathwater. He argues that because animal experiments don't always yield reliable results, animal research must therefore be halted. Dr. Greek's views serve the animal liberationists who support him. I don't believe they serve any human being who is prone to disease. That's each and every one of us. I came to Amherst from Chicago a little over a year ago to head a new division of the Center for Inquiry called the Commission for Scientific Medicine and Mental Health. The commission was established to defend science-based medicine and mental health practices against the attacks of medical quacks and other opponents of evidence-based healthcare. We sponsor 2 peer-review journals, the Scientific Review of Alternative Medicine and the Scientific Review of Mental Health Practice. So why am I here defending the use of animals in biomedical research? I'm here because animal experiments are an essential tool for biomedical studies. This vital tool is now being attacked not by researchers, but by spokespersons for animal liberation groups such as PETA, that's People for Ethical Treatment of Animals, and its front group, Physician Committee for Responsible Medicine, which is neither a committee of physicians nor an advocate for responsible medicine. Today, science is coming under increasing attacks from a wide variety of groups that have agendas inconsistent with free inquiry and reason, which are what the Center for Inquiry and the Commission for Scientific Medicine and Mental Health were established to defend. However, today I am speaking only for myself. I am not speaking for either CFI or the Commission, because the Commission has not yet adopted a position on these issues. I therefore speak only for myself. While I reported on the animal liberation movement 10 to 15 years ago for the Journal of the American Medical Association, I haven't kept up with the issues or on the escalating acts of violence against scientists who use animals in their research. Less than 2 months ago, Dr. Novus gave a talk at CFI, which brought me back to this subject and to agreeing to this debate. I've been working on it in my spare time, and I regret that the talk is only half finished. so you'll have to forgive me later on when I have to wing it. What motivated me most were the attacks on the March of Dimes that Dr. Novus, Dr. Greek, and other animal rights extremists are conducting, such as they do in the publication like this magazine, published by the Peter-sponsored Physicians Committee for Responsible Medicine, or PCRM. A few weeks ago, on April 12th, I and many others throughout this world celebrated the 50th anniversary of the conquest of polio. On that day in 1955, March of Dimes funded researchers announced the results of the Salk polio vaccine field trial. The whole world rejoiced when it was announced that the vaccine was safe, effective, and potent. That was just 17 years after President Franklin Roosevelt established the National Foundation for Infantile Paralysis in 1938, which became known as the March of Dimes, following a comment by comedian actor Eddie Cantor, who told the radio audience that he wanted to see a March of Dimes right into the White House to fight this terrible disease. And March those dimes did. It took the contribution of more than 100 million Americans to do it. But in less than 20 years, March of Dimes-led researchers developed 2 vaccines, not just one, to conquer polio and wipe it from our planet. But they had to do it with the help of tens of thousands of monkeys and other animals, which is why animal rights extremists now despise the March of Dimes. But let's look at what the sacrifice accomplished. 5 years ago, I don't have the slides. 5 years ago, only a dozen or so countries were reporting cases of polio. And last year, that number shrunk. I had slides, but apparently they're not here. There's only a handful of countries now where polio is isolated. India, some parts of Africa. Thanks to international efforts to eliminate the remaining pockets of polio with the polio vaccines, this dreadful disease is close to being completely eradicated the way smallpox was. I would say there's probably only one other person here that remembers what the terror of polio was like. Many of you probably don't know anybody who was crippled with polio. Few of you can ever imagine the terror that this disease caused. One of the great polio epidemics struck New York City in 1916, which caused thousands of people to flee the city in a panic, only to be met by armed citizens in the neighboring communities who threatened to shoot them if they didn't turn around and go back. The epidemic of 1952 was far worse. It crippled and killed more than 58,000 children and adults in the United States alone. And the following year, in the fall of 1953, the March of Dimes began the largest clinical trial ever conducted. More than 1 million skilled children were vaccinated with the killed virus vaccine that Dr. Jonas Salk developed by using 17,000 monkeys to establish that that vaccine could produce immunity to every strain of polio virus in the world. Another 3 million children served as controls. I was one of those children. I was in the 2nd grade, and I remember seeing the volunteer children getting vaccinated in my school's gym. And I remember the news, a little over a year later, announcing the spectacular success of that vaccine. It's hard to describe the terrible fear that returned every summer, the season when polio was by far the worst. Parents wouldn't allow their children to go to swim in public pools. Like my parents, many wouldn't let their children go to movie theaters. I was 10 years old before I was first allowed to go to a movie theater. And I remember that day. I saw James Cagney in A Man of a Thousand Faces, in case you were wondering. Excellent film. And it's not just a disinformation campaign that animal rights extremists are conducting about the history of the polio vaccine. In fact, we saw a slide here saying that the polio vaccine was delayed because of animal research. That's categorically not true. I am also dismayed by their attacks on the Foundation's current efforts to defeat birth defects. After conquering polio, the March of Dimes rolled up its sleeves even higher than it did before and turned its oppressive fundraising and research granting machinery to the goal of conquering birth defects, all several thousand of them. I know how impressive this machinery is because after graduating Columbia University Graduate School of Journalism, I went to work for the March of Dimes as a science writer. It was my job to study the research funded by the foundation and explain it to the news media and to the public. I worked for the foundation for 3.5 years and came away with the utmost respect and appreciation for what this remarkable charity has done and continues to do. So when I learned about the campaign of disinformation that animal rights extremists are conducting against the March of Dimes, I volunteered to speak up here. Because the March of Dimes refuses to be blackmailed into discontinuing the funding of animal research, animal rights extremists are trying to persuade groups to stop donating to the foundation. Yes. Many of you haven't seen an iron lung. You may not even know what it is. But back half a century ago, hospital wards were filled with iron lungs that did the breathing for children and young adults. Today, they're relics of museums. This is a website that PETA has up, March of Crimes website, where it vilifies the March of Dimes and says what, well, I don't have a copy of a letter that Dr. Nathan Nogus, identifying himself as a representative of Physicians Committee for Responsible Medicine, sent to the executive board of the Spanish and Latino Students Association of the University of Rochester in an attempt to stop them from raising money for the March of Dimes Birth Defects Foundation. Quote, "You might not know that the March of Dimes sponsors exceedingly cruel research that in no way helps children," unquote, he wrote. Nathan referred the Spanish and Latino Students Association to a website run by PETA called the March of Crimes. PETA and its pseudoscientific front group, PCRM, are also attacking other leading charities that refuse to stop funding animal research, such as the American Cancer Society, the American Heart Association, the Arthritis Foundation, and others. For example, here's a sidebar published. Oh, by the way, this is a little handwritten note from the March of Dimes campaign coordinator of PETA that was left on the front stoop of the president of the March of Dimes. It's a little intimidation tactic. It mentions how she visited all the neighbors and told them what terrible things the president and the March of Dimes are doing. Well, you'll find out how intimidating this is when I talk a little more about PETA's operations and the people they support. This is a sidebar that appeared in that Good Medicine magazine of PCRM. It encourages people, instead of donating to the Arthritis Foundation, which conducts great research in the corporate better treatments and cure of arthritis. Instead, it says to donate to the Arthritis Research Institute of America and the Arthritis Trust of America. So let's take a look. Here's the Arthritis Research Institute of America. First, let me tell you, the Massachusetts, do I have that? Yeah, here's the Massachusetts chapter of the Arthritis Foundation. It has a 3-star rating. 86% of its donations that people give goes to funding programs. Now let's take a look at the charity that PCRM is recommending. 35.3% goes to programs, the rest goes to salaries, expenses, and fundraising efforts. Let's take a look at the other organization that they're telling people to donate to. The Arthritis Trust of America. This is a fringe group that claims to have already found a cure for arthritis and other autoimmune diseases. Its websites state, quote, "We tell folks how to get well from so-called incurable autoimmune or collagen tissue diseases, such as the rheumatoid diseases and related diseases, by means of physician referral, publications, and when money is available, refund alternative, complementary, holistic medical research." Of course, if this foundation does have the cure for so-called incurable arthritis, why would they spend any money on research, alternative or otherwise, when they should be using it to provide the cure to sufferers? So is this responsible medicine that PETA and PCRN is offering? I'd like to tell you more about PETA and the so-called Physicians Committee for Responsible Medicine. But I first want to tell you about some of the Marchadyne-supported research involving animal studies that have already saved hundreds of thousands of children from death and crippling birth defects. Respiratory distress syndrome, also known as hyaline membrane disease, was a great cripple and killer of low birth weight babies. Babies born before their lungs are mature enough to function normally would often develop this mysterious, devastating disease that would cause their tiny lung sacs to collapse, leading to suffocation. Marchadyne-supported research with animals led to the development of a very effective treatment that has cut the death rate from respiratory distress syndrome in half. It also greatly reduced the incidence of lasting disabilities among survivors of respiratory distress syndrome. The treatment involves coating the baby's lungs with either an animal-derived or artificial surfactant until the lungs mature enough to produce their own natural surfactant. This research was conducted on animals first because you can't do this with human babies. Another great killer of newborns was a birth defect called persistent pulmonary hypertension. This defect causes increased blood pressure in the blood vessels of the baby's lungs and prevent the lung sacs from expanding and contracting normally. A major advance in treatment of babies with this defect came about through Marchadyne-supported animal research. The treatment involves a tiny amount of nitrous oxide, a powerful but toxic gas that causes constricted blood vessels to relax. The safe and effective amount of this gas for babies was worked out using animal models for the disease. The treatment has significantly reduced the total of injury and death from this birth defect. Patent ductus arteriosus is the most common birth defect involving the baby's heart. Before birth, there is an opening between the fetus's aorta and the pulmonary artery to circumvent the lungs, which of course aren't used in utero. In some babies, that opening doesn't close after birth, which prevents the baby from getting sufficient oxygen through his or her lungs. Severe cases had to be treated with risky and traumatizing surgery. Thanks to Marchadyne's funded research involving animal models, the causes of the birth defect were worked out and effective drug treatments were developed to safely close that opening. Animal research showed how prostaglandins keep this opening from closing after birth. And animal research showed that prostaglandin inhibitors could safely close it. So now drugs like indomethacin are being used to treat babies with patent ductus arteriosus instead of hazardous heart surgery. That same research led to a successful treatment for another common birth defect of the heart that once killed many newborn babies, hypoplastic left heart syndrome. Results when the left side of a baby's heart is too weak to pump enough blood through the heart to keep the baby alive. By treating those babies with a prostaglandin, doctors now are keeping the baby's ductus arteriosus open instead of closing it. Creating a patent ductus arteriosus in these babies allows the right side of the baby's heart to pump blood through the lungs to keep it alive long enough for doctors to surgically repair its malformed heart. These are just a few of the many advances brought about by animal research, the research that animal rights extremists insist do not help babies. This old photograph of a miner carrying a canary into the mine. They used to be used as an early warning for miners because they're much more sensitive to carbon monoxide poisoning than humans. So when a canary staggers and falls off the perch, the miners knew to get the hell out of there. Carbon monoxide is It's an odorless, tasteless, colorless gas. You probably know just how dangerous it is in the home, where it can't be detected if you don't have a device. Well, we didn't have carbon monoxide devices to test for carbon monoxide until recently. So they used canaries because, as Dr. Greek said, the respiratory system of the canary is quite different from humans. But a difference that makes no difference is no different. The fact of sensitivity, the fact that a canary is more sensitive than humans to carbon monoxide allows it to be used as a model, as a safety one. And that's exactly what many animal studies are doing. They are not, as Dr. Greek claims, they are not predictors of whether human beings, whether a drug is safe for human beings. They are models to tell us something about the drug, how it's metabolized, how the body of a mouse or a mammal uses it. It gives us the information that we can then use in developing the drugs. Steps in drug development. Preclinical toxicological tests. Preclinical, that involves toxicological tests, animal studies. Next comes phase 1 clinical trial, which requires about 20 to 80 healthy volunteers. Then there's the phase 2 clinical trial, which will use about 100 to 300 volunteer patients. Phase 3 clinical trials require up to 3,000, 3,500, or even more patients. Following the completion of phase 3 trials, the FDA will review the data submitted. The animal studies are submitted to the FDA, and they use that too in order to see things about the drug, such as carcinogenicity and teratogenicity, meaning, does it cause birth defects? You can't it's not ethical or legal to conduct a carcinogenic test or a teratogenic test on a human being. So they're not done. So they use animal models, a number of different species, to learn something, to see whether these drugs might, not will, but might cause cancer or birth defects in humans. This is the canary in the mine. This is not not used as a test to see if these things cause cancer or birth defects in humans. This is a misrepresentation of the facts. They are guides to the clinical trials. To put it another way, animal studies of drugs are not used to determine whether these drugs are safe and effective in humans. They are done to generate hypotheses. The hypotheses are then tested in clinical trials using humans. This is a graphic to show you just how complex the drug approval process. But animal testing just collects the information that's useful for doctors and researchers to test them in humans. Accutane. Accutane is a vitamin A. It's similar in structure and function as vitamin A. The actual name is isotretinoin. Accutane is the product name, the trade name. Its insert warns very, very, very strong warning. It causes birth defects. It is a major, very powerful teratogen, causing about 20% of pregnant women who take this drug during the critical time will have childhood birth defects. This was not discovered in humans. It was discovered in animals. Or I should say it was predicted by animal studies. Vitamin A, high doses of vitamin A causes birth defects in animals. So when this drug came along and was about to be marketed, the FDA researchers knew they had to watch this very carefully because vitamin A, large amounts of vitamin A in the body, a small amount of it is turned into isotretinoin, which was a very powerful teratogen. Well, sure enough, they watched. And when the first defects, birth defects, started to appear in women who had taken isotretinoin, the alarm really went up and they started getting very serious about this warning. So again, the animal studies was a canary in the mine. It was not a test to see whether it's safe in people or not. Clinical trials are done to determine that. Vioxx. There's that article in the, all right, don't have it. There's an article in Good Medicine which blames the Vioxx debacle on animal studies. And Dr. John Pippin, a cardiologist, a member of the so-called Physicians Committee for Responsible Medicine. By the way, did I tell you the Physicians Committee for Responsible Medicine has very few physicians? Only 5% of the members are physicians. As often said, Physicians Committee for Responsible Medicine is neither a committee of physicians nor a committee that advocates responsible medicine. Dr. Pippin, the cardiologist, wrote, "Good science could save consumers from the next Vioxx, but that won't happen unless the government stops relying on antiquated animal tests." And he says, as a first step to keeping consumers safe, the FDA must stop pretending that animal tests accurately predict results in humans. If you take a look at the labeling, FDA-approved label that goes with Vioxx, and try to find anything in there about animal studies, except for teratogenicity and carcinogenicity, you'll find nothing, because the approval of Vioxx by the FDA was not based on the animal studies. It was based on clinical studies, phase 1, phase 2, phase 3. Animal studies use about 1,500 animals. That's the preclinical research that's necessary. Following that, as I said, there's the clinical trials. Up to 3,500 people. Now, the problem with Vioxx had nothing to do with animal studies, contrary to what PCRM and its propagandists have been saying. The problem with Vioxx has two parts. One is the advertising. In the old days, before drugs were advertised on television all over the place, it took time for a new drug to get out to the market. By then, doctors tended to see adverse effects. Now, 1 million to 10 million people are taking the drug within a year. So, lo and behold, if there are rare events, adverse events, they show up in large numbers, as it did with Vioxx. How are they going to detect a serious adverse effect that occurs in 1 in 10,000 people when they're only testing it on 3,500 people? That's the problem. There's also a problem with post-marketing surveillance. It's inadequate in this country. It's not required by the FDA. There's no real good system for it. That has to change. That's the problem with Vioxx. That's the problem with all the other drugs that Dr. Grigg talked about that pulled off the market. Had nothing to do with animal research, because the clinical study, the studies that the FDA required, were done in people. With the exception for testing for cancer and birth defects, because you can't do that with people. Although some people now are arguing that we should. I do not agree. One of the things that Dr. Grigg says over and over is that 20% of, what was it? When drugs are tested, when they enter clinical trials, only about 80% of them fail. Only about 20% get through the approval process. And he blames animal studies, claims that's the problem, because the animal studies didn't show that 80% of the drugs have serious adverse effects with humans or may not be effective. Well, he's not discussing the other numbers. The other numbers are this. When drug companies are trying to develop a drug, they'll go through 5,000 different compound screening. Only 250 will make it into the laboratory for the preclinical research involving in vitro studies and animal studies. Out of the 250, the in vitro studies and the animal studies will show that 245 of them, 98% of them, are ineffective or too toxic. They will not be used on human beings. So that's the number that he doesn't like to confront you with. Yes, 80% of the medicines, new drugs, that enter clinical trials don't get through. But that's not because animal studies fail. What the animal studies did was it highlighted, it told the researchers what to look for, what organs may be affected, so that in the clinical trials they watched more carefully, looking for signs of toxicity. And that's the reason many of these drugs do not get through phase 1 or phase 3 or later on phase 3. Science learns by making mistakes and then correcting them. What opponents of science love to do is to shift through the scientific record and pull out studies that were wrong, while ignoring or even denying those that were right. Dr. Greek unfortunately does this. He's filled 3 books with things taken out of context, things misconstrued, and things just plain falsely stated. I am going to jump to some examples. Before I do, I wanted to say, tell you what Dr. Thomas Starzl, the noted pioneer for transplantation, said about animal studies. He said about his own pioneer work for organ transplantation, he said that the first study he did, most of the subjects died. He learned from that and adjusted his technique. And the second round of studies for the transplantations experiments he did, most of the subjects actually survived. The majority did. The third set of experiments that he conducted, there was only 1 or 2 deaths. Nearly all the other subjects survived. His fourth experiment, every one of the subjects survived. The difference, the most important difference between these studies was that the first 3 were done in animals. The fourth one was done in babies. But Ingrid Newkirk, co-founder, president of PETA, says, there's no rational basis for saying that a human being has special rights. A rat is a pig, is a boy, is a dog. And yeah, it's Dr. Starzl. Let me get to this. PETA's spokesman, Bill Maher, I love his politics, more or less, but he's a staunch spokesman for PETA, and he conducted a campaign against Columbia University recently, calling on them to stop their Frankensteining of primates. Unfortunately, Maher doesn't believe in vaccination either, he says. That's another theory that I think is flawed, and that we go by the Louis Pasteur theory, even though Louis Pasteur renounced it on his own deathbed and said that Beauchamp was right. It's not the invading Germans, it's the terrain. David, did you know that Pasteur said that on his deathbed?
I was unaware.
Yes. He doesn't believe in the germ theory disease, and he says, to those people who say, my father is alive because of animal experimentation, I say, yeah, well, good for you. This dog died so your father could live. Sorry, but I am just not behind that kind of trade-off. Well, that's what Peter, and that's what PCRM is all about. PCRM is a front group. It's funded and it's basically run by Peter. Physicians Committee, the AMA in 1991 censored the PCRM, saying the Physicians Committee for Responsible Medicine has been formally censored by the AMA for purposely misrepresenting the critical role animals play in medical research. I wanted to make a comparison, but I'm running out of time, so I'm going to rush ahead. Between some of I spent a lot of time reading through Dr. Greek's books, and I tracked down his references, and I found so many examples where he left things off, misquoted, misrepresented, distorted, or made things up. He quotes Stephen Schneider, this noted biologist, environmentalist, saying each of us has to decide what the right balance is between being effective and being honest, as a way of showing how some scientists are a little less than honest. Only he left out the last part of the quote. I hope that means being both. In one of his books, he has a reference to show that research of dogs did not lead to information, knowledge about diabetes. He quotes some obscure publication. He says, in 1895, Hod Ponsonby reviewed the literature and found 72 cases of diabetes accompanied by lesions of the pancreas. Well, several years before, there were experiments done on dogs where they removed the pancreas and the dogs became diabetic. So I don't know what this reference is supposed to mean. This is not legitimate scholarship. Yeah, that was that. Okay. Dr. Greek, in one of his books, says that there has been no, despite all these animal research, has been no improvement in the overall mortality, the adjusted mortality of cancer in humans. Well, let's take a look at the facts. Look at the cancer incidence and death rates in children. The incidence from 1975 to 2001 has gone up, but look at the mortality, dropping. Adult males, even lung cancer is taking a sharp turn beginning in 1990, and most of the other cancers are going down. Adult females, likewise. You can see how lung cancer went up, but it's flattened out and it's on its way down, as most of them are. And if you look at combined cancer death rates, it's dipping down. What Dr. Greek said is simply not true. Perhaps he doesn't think that there are people like me that are exactly going to check these things out. Nicotine withdrawal symptoms were scientifically confirmed in humans in the 1970s. Nevertheless, rats were still being studied for signs of nicotine withdrawal in 1994. Well, I looked up that reference, and it wasn't to see if rats can become addicted to nicotine. What it was it was a test to see if a drug called I can't say this right, thank you, whether it can interfere with nicotine addiction. And that was done in 1994, and as a matter of fact, a year later, they moved on to clinical trials in humans. That representation that Dr. Greek did, he took, he didn't think people was going to go and look that reference up, well, I did, and didn't say what he said it did. Okay, listen to here's a debate in 2002, between Dr. Greek and Dr. Robert Spaff. It doesn't work. Well, Dr. Greek corrected, or Dr. Spaff said that many veterinary medicines are human medicines because of the similarities. Human beings and animals or pets share about 200 of the same diseases. Well, Dr. Greek said, well, they were using human medicines because no pharmaceutical company will invest any money to develop drugs and that they don't have to. There's no money in coming out with new drugs. Well, just look at all the current dog vaccines available. There's something like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, about 11 different vaccines that are just developed for cats and dogs. And this is something that's new. Now, Dr. Greek's wife is a veterinary doctor. Why doesn't he know this? Here's a new tablet for dogs, 1 a month, and it prevents fleas. It interferes, as a new novel drug, interferes with chitin, formation of chitin. Insects use, their exoskeleton is made out of chitin. The flea drinks the blood, gets the enzyme, the inhibitor, can't make chitin, the eggs are sterile. And they've got an injection once every 6 months for cats. Now I can go on with numerous other examples of how he's misrepresenting the facts. This is not science. This is, oh, here's this was a good one. Maybe I'll get to it. Oh no, I don't get a rebuttal. But maybe the question and answer period. And I can't play sound. Well, I wanted to play you what Dr. Jerry Vlasic, associate of Dr. Greeks, said at an animal rights conference, where there's an incredible, chilling soundbite where he's recommending the assassination of animal researchers. Not lower ones, but the higher up ones. Send a message to stop this. Well, Dr. Vlasic has been a partner of Dr. Greek, and in fact, he was your science advisor, I believe. And in fact, as of last summer, your newsletter has him listed as a science advisor. This man and his wife have been barred from Great Britain because he's a danger. I don't have time to show you all the other people that are associated with Peter, that work for Peter, that work for PCRM, who are involved with arson, who support arson, violence, intimidation. This is all a plot to stop people from abusing animals in their minds. Thank you.
Dr. Greek, you now have 5 minutes for a rebuttal.
Thank you very much. Well, first let me thank Mr. Skolnick for agreeing to participate in this debate. As I listened to his arguments, though, I got the feeling that we were attending 2 different debates. To begin with, I represent Americans for Medical Advancement, okay? We are an organization that is a patient advocacy group. We have in our mission statement, which you can find on the internet, that if animal experiments cure humans, we're all in favor of it, okay? The board is comprised of 6 individuals, 5 out of 6 eat meat, wear fur, and hunt. So to confuse us purposefully or not with an animal rights group such as PETA or PCRM, I think is slightly disingenuous. We oppose all illegal activities. We don't just oppose violence, we oppose illegal activities in mass. We oppose the whole thing. Yes, Dr. Vlasic was on our scientific advisory board, and he was recently fired. So, as a matter of fact, we fired him before he made his outrageous statements about violent activities because we thought he was going in the wrong direction. So if you can condemn us because of past company that we have kept and fired, I guess that's up to you. The other theme, I guess, if you will, is that we're somehow part of the animal rights movement. Again, when 5 out of 6 of your board members eat meat and wear fur, I don't see how you can get that. Well, let me just say this. If we were to veto every idea that had a fringe or lunatic supporter, no idea would ever be taken seriously. And I mean, clearly, a lot of the animal rights movement love what it is that we do. I wish they loved us enough to donate their money, but they don't. One reason the vested interest groups choose invective instead of explanation is that they have no scientific support for their position. Now, you'll notice that I did use references in my talk. For example, when I talked about the National Cancer Institute saying they lost cures for cancer, that had a reference in Science, which you can go and look up. And our books have over 1,000 references to peer-reviewed scientific journals. And I implore you, please, look them all up and come to your own decision about whether or not we've been honest. I stand by every statement we've made. By using the phrase animal rights activist in the same diatribe about animal models, vested interest groups seek to avoid engaging in true debate and instead imply that those who disagree with them are anti-science, inflexible, irrational, anti-human, and so forth. With regards to the polio vaccine and some of the other things that Mr. Skulnick said, we had some disagreements, but not really. No one seriously claims that animals were used as causal analogical models in the development of the polio vaccine. Where they were tried as CAMs, they failed. For example, one vaccine was developed in the 1930s based on using monkeys as CAMs, and children ended up losing their sense of smell for the rest of their life. I've actually met someone who was involved in that study. In terms of them, animal models delaying the polio vaccine, I wasn't alive then, okay? I have no first-hand knowledge. Al Sabin, I think, has first-hand knowledge. And again, if you go to our books or our website, you will find a reference from the Congressional Record, okay, where Al Sabin testified under oath before Congress, raised his right hand and said, quote, or I shouldn't say it's a quote, he said that animal models delayed the polio vaccine for decades. Hey, if he says it, I believe him. Now, when the polio vaccine was finally produced, you remember in numbers 3 through 9 that I had on the board, you can use animal parts. And that's how the polio vaccine was produced. It was produced using kidney tissue from monkeys. I mean, I'll give you that. That's not our argument. Our argument is that you cannot use them to test drugs and study human disease. Just a couple of things. We don't give nitrous oxide to babies, Mr. Skulnick. Nitrous oxide is something that I, as an anesthesiologist, give to people who don't want to be awake anymore. We give them nitric oxide. Nitric oxide was, again, an example of something that was discovered in the aorta of a pig. Now, if you don't know the difference between nitrous oxide and nitric oxide, that doesn't impress me. Cancer mortality rates. Yes, cancer mortality rates are coming down. Not because of treatments, because of two reasons. Number one, people are stopping the habit of smoking. And number two, we're diagnosing their disease earlier. We're not curing them. We're not curing anybody of cancer. I shouldn't say this is a sweeping generalization, but by and large, we're not curing cancer. I mean, I don't think I need to defend that point. This really gets down to the canary in the coal mine. I think that's an excellent example. In the 1930s, if I was going into the coal mine, I'd want that canary on my shoulder. I wouldn't want him on your shoulder, because we might be 10 feet away from each other. So using the canary in 1930 was a very viable thing to do. Today, if I went into the coal mine, I'd want a transcutaneous PCO2 monitor, a PO2 monitor, and a PCO monitor. And so that's what you can do. In the old days, we used canoes to cross oceans. And you still can. I mean, that's what the look you're going for. Good luck to you. We can also use the Concorde, or at least we could until they retired it. Phase 1 clinical trials, the actual number is 5 out of 100 drugs that pass safety tests in animals and it go on to phase 1 clinical trials in humans. 5 out of 100 make it to the market. Now, I don't care, we can have an argument about what are animals used for in drug testing. But if your batting average is 5 out of 100, man, that's not good. That's not good if you're playing hockey, okay? So that's just not a good thing. Lastly, if I can get through it, Mr. Skulnick did show us some so-called victories from using the animal model, but these are really a Potemkin village. It's an impressive facade, but it hides an undesirable fact or state. Now, he did present some interesting, albeit misleading anecdotes, but the plural anecdote, even if these anecdotes were true, is not data, and science is concerned with data. And I think you'll notice the difference in our two presentations. I mean, I gave you a very scientifically oriented, probably over somebody's head, some of it was even over my head, debate. So the question when you use anecdotes is this a fair presentation of the general case? And what I tried to do is give you the scientific theory along with some empirical data from peer-reviewed journals that supported my case. I don't think my opponent did that. Also, in arguing for the importance of animal models, he used a shotgun strategy, and I don't have time to pick out everything that was wrong with every one of them. I just don't have the time. But to give a couple of examples where a certain animal species reacted similarly to humans, And from those limited examples to conclude the animal model as a paradigm is viable is the fallacy of loaded conclusions. It would be like profiling several lottery winners and concluding that playing the lottery is the best way to save for retirement. Thank you.
Okay, thank you. I have a question for Ray Greig, and when I was listening to Andrew Skolnick's presentation, one of the things that spoke to me a bit is that animal trials are not used to prove the efficacy. I think this was what his position of, but they're used to narrow down or to perhaps weed out what trials should go into clinical trials, and it's really clinical trials that prove the efficacy of drugs. And I wonder if you could speak to that and clear that up or say how that's relevant to this.
Sure, thanks for the question. Animal trials, if you read the drug company's propaganda, are done for one reason and one reason only, and that's to save the general public from death, okay? And even if you read Nature Review Drug Discovery, Nature, biology journals, and so on, they're very about that. So I don't know where this concept came from that we test on animals, use vis-a-vis drug testing to get an idea or to use it as a heuristic. If there's one place that animals are used as cams and are meant to be predicted, it's in drug testing. Now, where the confusion arises sometimes is that the animal testing stage is sometimes done first, and that's when we talk about phase 1 human clinical trials. And that's the data that I talk about when I say 100 drugs enter phase 1 clinical trials and 95 of them fail. That's really what animal testing is for. It's to protect those people in phase 1, 2, and 3 clinical trials. Okay? Now, they would also like it to protect the people in the general public when the drug is marketed, but it's a distinction without a difference. Because if you feel safe taking a drug or that was part of 100 that got weeded down to 5, I mean, that's just not a good batting average. And you have to flip that argument on its head. Remember, the National Cancer Institute says we think we've lost cures for cancer. Well, I think Mr. Skolnick's data is roughly correct. You know, you do get 250 drugs to animals, and from the animals, you know, you're left with 100. What makes you think those other 150 weren't a cure for breast cancer? You don't know. I mean, if the animal model as a predictor is so bad that at best you're getting 5 out of 100 right, that's terrible. And that's not just my opinion. Mark Levin, the CEO of Millennium Pharmaceutical, showed a real nice slide one time where he tested drugs on rats and on humans, and there was no predictive value whatsoever. It was like a coin toss. It was hepatotoxicity. But we can go into that further later if you want.
Well, let me show you why we do and why we're required to do preliminary studies on laboratory animals before we do the studies on people. Here are some of the pictures that became evidence in the Nuremberg trials of the Nazi doctors. You may be surprised to know that the only modern country to totally outlaw animal experiments, animal studies, was Nazi Germany. Adolf Hitler in 1933 outlawed it. I'm not saying that that's the reason he turned to humans. He had other reasons to turn to humans. These were people whose lives were not worth living. These were the infirm, the mentally retarded, the handicapped, the Jews, the gypsies, the war prisoners, and anyone else that got in his way. Hyperthermia experiments where these Nazi doctors submerged people in ice water until they died. Then they used to do research for pilots. Their Luftwaffe pilots bailing out at high altitudes. They put prisoners into decompression chambers. And this was a cute one where they just starved the women and then removed their genitalia, the ovaries, and sent them to the universities for study. Out of the Nuremberg, out of the Nazi Holocaust and the Nuremberg trial came a code that the judges at the Nuremberg trial drew up that defined what is ethical for human experimentation. Number 3 states, the experiments should be so designed and based on the results of animal experimentation and a knowledge of the natural history of the disease or other problem under study, that the anticipated results will justify the performance of the experiment. And from the Nuremberg Code, which was written in 1949, in 1964 and modified several times, the latest in 1983, and accepted by the World Medical Assembly, which represents the medical societies of over 80 nations. They drafted the Helsinki Declaration, moving, I don't know if I have it now. It moved the rule to number 1, all human experiments to be ethical must be done based on a thorough review of the medical literature, the scientific literature, and after properly conducted animal studies. That has been the basis of our laws and the laws of most countries. That's why we do the studies. Dr. Greek in his book says, well, it's the greedy pharmaceutical companies, they don't want to be sued, so they're going to do these animal studies so they can defend themselves in court. Well, that's nonsense. Anyone who follows litigation with drugs knows that toxicology studies, preclinical studies with animals will not save the drug company. A great example is Bendectin. This was the most well-studied drug in pregnancy. Am I out of time? Oh, I'm sorry. You have to, wait. That's all right.
Other questions?
I take back everything that I said after the time.
For Dr. Greek, my question is regarding polio. Could we have come about with could a cure for polio have come about without animal experimentation? And if so, how long would it have taken?
Thank you. Well, again, according to Dr. Sabin, it would have taken 30 or 20 or 30 years less long than it did. Okay? Again, that's not Ray Greek, that's Al Sabin, who probably knows a thing or two about it. What animal experiments allowed us to do vis-a-vis polio was it allowed us to grow the virus, and it allowed us to test the purity of the vaccine. Okay? And again, if you go back to my second slide, where I talk about how animals are used in science and in research, animal tissue today is used ubiquitously to grow things. It's part of culture media. Now, we don't have to use animal tissue. We can use tissue from humans, of which there's a surplus. People die every day. Society finds that a little unsettling, and that's fine. But there's a difference between something being necessary and something being functional. And so the original polio vaccine was grown using kidney tissue from monkeys. Okay, fine, great. I mean, that's fine. What paved the way for the polio vaccine, you gotta remember, Sabin and Salk didn't win the Nobel. Okay, the guys who won the Nobel were the guys who made the virus grow in vitro. That's what took the brains. Okay, I'm not saying Salk and Sabin were dummies, they're far smarter than me. But that's where the great breakthrough occurred. So it's disingenuous to say we studied polio in monkeys. No, we didn't. And when we did, we thought that polio entered through the nose and went to the CNS. That's what Sabin was talking about when he said it delayed the vaccine for decades. That was wrong. It went through the gut. Okay? So, we can't second guess history, but all of the ingredients were there many years before they were all used.
All right, please forget what you just heard. Sabin had been up to his dying day, had been very upset about how his testimony before Congress had been taken out of context and has been misused. He always defended animal research as having been essential for his work and for Sabin's work, Jonas Salk's work. The mistake that happened, by the way, it wasn't because of the animal studies that an error had been made. The error had been made Not because they used animals, but because it was misunderstood. There was plenty of evidence already, going back quite a while, that showed that the gut did get infected. What happened was Sabin, in the late 30s, had conducted an experiment on fetal human tissue, not animals. He got a fetus, and he made cultures of the cells for different organs of the fetus. It was an aborted fetus. And unfortunately, he got a strain of the vaccine. Remember I said there's well over 100 strains of poliovirus that was expended throughout the world? He got one that was a laboratory specimen. It was grown and grown and grown, not in nature. It was grown in a laboratory for many, many generations. And it lost its ability to infect the gut. He didn't know that. So he used this strain from Rockefeller University and it did not infect anything but nerve tissue. It was an unlucky break. He could have used 100 different strains. He picked one that didn't infect nerve tissue. It was a mistake that set them back for about 14 years. Well, anyway, Dr. Greek blames that on animal studies. It had nothing to do with animal studies. In fact, it was grown in human tissues.
Perhaps I can also interject here. There was also some enmity between, or differences of opinion between Salk and Sabin. I knew Jonas Salk, and the difference of opinion was the use of inactivated virus versus a live attenuated virus. Sabin believed strongly in the latter, and Salk believed equally strongly in the former. Salk got there first, and that was the first vaccines that were used. And Sabin was in an unforgiving mood after that because he thought that Salk had got the priority for that. So there's an element of a scientific controversy. It's a fascinating history of two great men.
Could I ask for clarification? Because what I heard
Dr. Greek say is that Sabin said something about animal experimentation. What I hear Andrew Skolnick say is that Dr. Greek made a statement about animal experimentation. And what you're saying by saying that is that Sabin didn't say that, and what you're saying is that Sabin said that. Yes, what you said was that what Dr. Greek said this about animal experimentation, and Dr. Sabin believed and said something else, but what you said was that Sabin said it. So I just like a clarification of what did Sabin say.
His quote was accurate.
Yeah, Sabin said it. And that's all I'm saying. To get into how the polio vaccine came about, you know, we could go round and round on every, not just the polio vaccine, but every aspect of it. And I tell you, just to hopefully speed this debate along a little bit, look, for the sake of argument, I'm willing to give the animal experimentation industry every discovery up to the year 1990. Let's just assume for the rest of the day that every breakthrough in medicine up to the year 1990 was 100% dependent on animals. I don't care. That's not the point. Even if that were the case, the point is that in the year 2005, Status Post the Human Genome Project, Status Post the discovery of complexity theory, Evo, Devo, and so forth, folks, the name of the game has changed, okay? So we can debate history till everybody's blue in the face, and it's not relevant to the argument that I am making.
I didn't say, Sabin said that, but it was taken out of, it was, you know, he was speaking before Congress, it was taken out of context and misused. He had rebutted the misuse many, many times, and Dr. Greek had been misrepresenting what was said, not misquoting. Anyway, the 1990, speaking of 1990, in Dr. Greek's third book, he has a list of the last 20 years of Nobel Prizes. He leaves out 1990. When he was debating Dr. Speth on radio in 2002, Dr. Speth talked about kidney transplantation being essential using the dog model. Dr. Murray had won a Nobel Prize for that. Well, Dr. Greek said, oh, Dr. Speth, you should know better than that, something to that effect. The first successful transplant was in humans. Was only successfully done in dogs 5 years after. Dr. Speth was dumbfounded. didn't know enough to contradict him and say that's not true. Unfortunately, it was not true. Might be why Dr. Greek did not put the year 2000 Nobel Prize in his book. Not 2000, 1992, because if you look at the 1992 Nobel Prize, it was to Murray for doing the first successful human transplant after developing the procedure in dogs. Check out, go to the website, if you doubt this, go to the website, look up the Nobel.org, and look up medicine, 1990. Dr. Murray had worked on dog model to perfect his technique.
Further questions or comments?
The question I have is for Mr. Skolnick. For me, in terms of the issue going forward, what I'm most interested in terms of the two different viewpoints here, is the predictability of one model or another. Do you have available any statistics in terms of the predictability in favor of using animals as models for disease or animals as subject for testing?
I don't have data to answer that question, but I do have this observation. There's little predictability, there's inadequate predictability. When they do a clinical trial of 5,000 people here, there's inadequate predictability of how the drug is going to work in the marketplace. Let's put it this way. I grew up where peanut butter and jelly sandwiches being served in school. Well, there's 3 million Americans who would possibly die eating the peanut butter and jelly sandwich. The 6+ billion people on this planet are very diverse, and you have atypical reactions. Animal studies are not perfectly predictable, it's not that accurate as a prediction, but that's not what animal models are mostly used for. They're used to give insights and generate hypotheses for clinical testing.
If that were the case, then a vast majority of grant applications to the National Institutes of Health would not end with these words. And we hope our proposed study will lead to a cure for X, Alzheimer's, stroke, Parkinson's, whatever, okay? What this debate is in large part about is about what I consider to be the very disingenuous attitude on the part of animal modelers and their representatives who say, on the one hand, this isn't about predictability, because they don't have the data. Okay, I've got the data that shows that animal models are not predictive. No, what this is about is using animals as heuristics. And I'll grant you, if you want to get a new idea, you can do that in a variety of different ways, and one of those ways is to study animals. But if you're going to take the American taxpayers' money, and if you're going to take the March of Dimes money, and if you're going to take these other charities' money, then be honest and say, you know, I'm looking for new ideas. And look, there's nothing intellectually inferior about looking for new ideas. I've probably never had a new idea in my life, okay? But let's not be disingenuous and say, when we're looking for new ideas, oh, we're really looking for the cure for cancer, because that's the only way they get the grant money. And again, what you're hearing here today are a lot of facts being thrown back and forth, and you're hearing a lot of just, you know, completely opposite statements. And I would encourage you to read the books. I don't make any money from the books. All proceeds from the books go to AFMA. But read the books, look up the references if you have the time. But if you don't have the time to do any of that, just, you know, use some common sense. Think of the things that I said during my portion of the debate that are kind of theoretical and sciencey and somewhat ethereal. But that's the core of the argument.
Could we have another round on this question?
Yes.
I could give some examples of very important predictability. Let's take a look at 1960s and farmers, turkey farmers in England had just devastating loss of turkeys. They were dying of what they didn't know what to call it, but they called it turkey X disease. Turns out what it was, after much research, using animals, using birds, It wasn't just turkeys, ducks, and other fowl got the disease. What it was, their liver was destroyed by a liver toxin. That was produced by a mole that was growing on the feed, aflatoxin. Well, sure enough, epidemiological studies later found that aflatoxin is a human carcinogen. It is causing a large number of liver cancers, primary liver cancers in humans. And it's something that now they're trying to keep out of our food supply. Now, another more recent, in the 90s, it's about 10 years ago, they found that cyanobacteria, blue-green algae, produce a toxin. And this toxin is liver toxic in animals and carcinogenic. And lo and behold, they did epidemiological studies in China, Africa, and other countries where the main water supply comes from surface water. And it is carcinogenic in human beings too.
Mr. Skolnick, if I could comment, I interpret those kind of examples as number 7 on one of the very first slides. I interpret that as animals as a modality for ideas, as opposed to what I was asking about in terms of animals as test subjects.
Please let Dr. Green comment
Oh, okay,
first,
sorry.
and then you have a chance to follow up.
I think if you take nothing else from this debate, take this example. Yes, there are animals who get cancer from the same chemicals that give humans cancer. There's no question about that. But when a scientist says the word predictability or predictive, that has a very specific meaning. Now, the last time I checked, and these numbers undoubtedly have changed, but the last time I checked, there were over 2,000 substances, chemicals, that were found to induce cancer in rats and cats and dogs and mice. You know how many of those substances cause cancer in humans? Last time I checked, it was 30. Let's say it's a double. You know, that's not predicted. You know, to say, I could go out and I could bet on next year's World Series, and I could place a $10 bet on every single team that's playing in the National and American leagues, and I'd be right. One of those teams would win, but I did not predict the winner. Okay, in science we have things like sensitivity and specificity and so on and so forth, and that's what prediction is all about. And I said, I think in my first talk, I said, you can almost always go back in time, retrospective, and find some animal that will react to a chemical or drug or disease or whatever, pretty much like the human does. That's retrospective analysis, that's not predictability.
One more round with us, or you want one minute each?
What I said was that they didn't go back and test turkeys for aflatoxin toxicity. They didn't go back and test animals to see if they also get cancer from pond water. These were the canaries that alerted scientists of the problem in human beings. They predicted. Now, I said in my talk that animal research is not primarily used to predict human reactions. They're used to provide insights and generate hypotheses that are tested in the clinical trials. Now, we can't give people suspected carcinogen. Dr. Green kind of misrepresented. We know there's about 90 pretty strongly linked, pretty strongly implicated carcinogens for humans. Maybe 30 that have been definitely confirmed. But there's thousands that are suspected because of the animal studies. We can't do the studies to find out if they are indeed carcinogenic in humans. It's unethical. So there's nobody, except maybe Dr. Green, thinks there's only 30. There's a hell of a lot more. The same thing with teratogens. Many suspected ones, only about 20, 30 that are known to be teratogenic in human beings, causing birth defects. We can't do those studies. We can only infer from the animals because we have no better means.
It's actually easy to do those studies. It's called epidemiology. And yes, there are only 30 confirmed. And again, maybe it's 40, maybe it's 50, 60, it's irrelevant. The point is that if the turkey is going to be predicted for carcinogenicity testing, what you have to do is that you then have to take those known human carcinogens, all 30 of them, give it to the turkey and see how the turkey fares. And the turkey doesn't do well, okay? That's what drug testing is all about. You have an animal that quote, according to Mr. Skolnick, predicted thalidomide's toxicity. Okay? Thalidomide was a drug that was toxic to humans when given to pregnant moms, and we did find the white New Zealand rabbit reacted the same way. Well, guess what? The white New Zealand rabbit was then exposed to a lot of other known human teratogens, and those known human teratogens passed with flying colors. That's the difference between predictability and retrospective analysis. The gentleman was right. You can get ideas from anywhere. And look, if I see cows dying in the field, I kind of want to know what they're eating. And then, yeah, let's see if other humans have eaten that, and, you know, let's explore it. It's a heuristic. I don't deny that animals, anything can be heuristic. But the problem revolves around what is prediction. What is predicted? And it's not analysis. It's not a casual relationship. It has to be causal.
Well, perhaps I could just interject. The original question was on predictability. I would direct you and perhaps other members of the audience who are interested to an article in Nature Reviews Drug Discovery 2003 by Abramowitz from Lexicon Genetics. What they did was take the 100 most popular clinically prescribed drugs, and they did mouse knockout studies in which you selectively delete a gene, and they asked whether the phenotype of that mouse knockout would have in fact been predictive of a model to develop that drug. And their conclusion, and you can study the conclusions as you wish, was that it would. So they would have argued, I suspect, for predictability. Any other further? You had one question. I'm looking at different questions. I'll give you the microphone to be easier.
You had stated in your presentation that humans die from medical research or drugs as a result of the human or the animal models that they use. But wouldn't that be able to be flipped easily, whereas if there were no animal models, then more humans could also die due to that?
Yes, thanks. I'll show my age and say that's the $60,000 question. There used to be a game show that was called that. $64,000 question. There you go. Right, that's what I'm arguing. I am not arguing that animals never get it right. I mean, that'd be great, because then, you know, if it caused cancer in the animal, it'd be safe and nuts. What I'm arguing is that they get it wrong more often than they get it right. And it's not black and white. It's not right, wrong. What it is it's misleading. I'll give you one example of that. In the 1980s, we knew that HIV was in the French blood bank, okay? The HIV tainted blood was being given to French citizens. The French government decided they weren't going to do anything about that because they knew that when HIV was given to monkeys and chimpanzees, it was safe. So the mode of transmission at that time of HIV had not been worked out yet. The result of reasoning from the animal model to the human was that about, I forget the exact number, 8,000 French people are now dead because they did come down with HIV, then AIDS, and then they died. So I think we have huge amounts of empirical data supporting the fact that animal models are flat out dangerous. Now, if you're going to say that animal models work, first you have to explain that in terms of evolutionary and developmental biology. You have to give me a theory why it should work. You can't just use isolated examples where animals and humans agreed.
But that's what you're doing in the same respect. You're giving isolated examples.
No, that whole first lecture was one long, boring theoretical science lecture.
And I agree with science, but I don't agree with what you're saying.
That's your prerogative. But when you accuse me of not just being up here presenting anecdotal data, I fail to see how that first 35 minutes contained a lot of empirical data, much less anecdotal. I mean, that was complexity theory, Evo, Devo, and so on and so on. And look, this is cutting edge stuff. You've got to know about Evo, Devo, you've got to know about complexity theory, you've got to know how all these things interact and relate. And if you don't know that, that's going to be problematic in terms of my theory. But the first 30 minutes of my lecture was theory. It wasn't even empirical.
Here we go again. The French, when the French prevented, well, let me back up a second. This representation of why the hemophilia patients in France got infected with HIV is false. The French did not allow, let me back up. The United States had marketed a test for HIV to make the blood supply safe. It was used in the United States and other countries. France would not allow it because its own company, the Pasteur pharmaceutical company, was working on one, and they asked the government to keep it out. They knew that you can get HIV infection from blood supply because it was already marketed in the United States and other countries, a test to prevent it. So this is just another misrepresentation. Page 81, Doctor, on his Specious Science, which is appropriately named, he says that while animal-based nutrition research typifies the ineffectiveness of animal model protocols, for instance, the need for vitamins A, D, and nicotinic acid, so powerful in maintaining immunity, was discovered in humans. No, it wasn't. All three vitamins were discovered with vitamin A dogs, mice, and dogs. You know what is really dangerous? Spurious science, false science, science that misleads and prevent us from finding more effective treatments.
Questions or comments?
We will take this question and one more question.
I was just wondering how, if Dr. Greek thought it was ethical, like how exactly you would give the drugs to the human population, what tests would need to be conducted if the animals aren't reliable, and if it's ethical to do that.
Thank you. That's a good question. Drug testing is actually easy. To begin with, I would do the same things that are being done now, which is a lot of in silico modeling, in vitro studies, and so on and so forth. Now, there are a lot of tests that drug companies could do at that stage that they don't do. Okay, and there's a movement within the drug companies themselves to do more bench work before the drug has ever progressed along. Second of all, I would skip the animal tests again, because the animal tests don't give you useful data. Now, if they give you useful data, then fine, use them. But again, if you read the books, look up the references, and so on and so forth, even the drug company today, in the journal, Nature Review Drug Discovery, is coming out almost every issue and saying it doesn't work. Okay? I mean, I'll take their word for it. And the other thing that I would do is going to cost the drug companies a lot more money, and they don't want to do this. And number one, when I exposed a patient or a volunteer in phase 1 human clinical trials, I'd use microdosing, spectroscopy, and PET scanning. And there's a really nice article, again, in the same journal, Nature Review Drug Discovery, a couple of years ago that outlines how that should be done. And the last thing that I would do is I would increase the number of clinical trials. I would make drug companies actually do tens of thousands. And I would also make them broader and longer, okay? And you can segue from, you know, if in year 1, if the drug looks promising, you can release it to some people in the population. And then you can release it to more and more and more. But that's going to cut profits, okay? And that'll never happen.
That may be the first thing I agree with Dr. Greek about, is that we need to slow down the dissemination of drugs once they've been approved. It's just ridiculous what's happening. I disagree with what he said, that drug companies want to do animal research. In fact, drug companies do not want to do almost any of these precautionary, slow, methodic studies. As a matter of fact, they're pressuring the FDA, and the FDA has actually put forth, this is hard to believe, on behalf of the drug companies, they have put forth a proposal that they will allow drug companies to test new drugs abroad among the developing nations without abiding by the Declaration of Helsinki. Look, the pharmaceutical companies are not doing animal testing because they want to. And quoting 1 or 2 drug company representatives saying, gee, we don't want to do animal studies that's not good enough science to show that you don't need animal research.
Perhaps worth also noting that the Environmental Protection Agency decided not to go ahead with its decision to study the toxicity of insecticides in children. They withdrew that decision.
But that was a political decision, that wasn't a scientific.
Well, it was a political decision for both, I think. One more question.
Thank you. I want to believe here that science kind of accommodates some level of false positive or false negative. That's when we talk about probability. Okay, my question in that regard to Dr. Gray is, could you kind of give us an idea of gain, what gain are we going to make if we stop animal experimentation, you know, in terms of drug development, either gain in time, gain in, have we been able to reduce the false positive or false negative that we are coping with now? And also to Mr. Skolnick, I would appreciate if you could also tell us what we will lose if we stop animal experimentation in terms of, you know, positive, false positive or false negative. Thank you.
Sure, I'll make this real quick because we're running out of time. What would you gain by eliminating animal testing? I'll quote the National Cancer Institute, Science 1997, we've lost cures for cancer because they didn't work in mice. Okay, period. And I will also direct you to the drug recalls of late, Vioxx, Celebrex, and so on and so forth, that animal testing did not prevent those drugs from coming on the market. Now the fallacy in this, we don't use animals to predict what's going to happen to humans, is this. The reason that drug companies have historically been able to do very abbreviated clinical trials, when they should be doing longer and broader clinical trials, is they have sold to the American public and to the FDA and to Congress, oh, well, we tested it on animals, so we already know it's safe, so we only need to test on a couple thousand humans. That's the dirty little secret that they don't want you to know.
So what was the question for me? Can you repeat your question, please?
What would you lose if you did not do animal studies?
You would lose a very valuable tool for gaining many important insights into how the animals, including humans, work, how chemicals interact with genes, how genes work. It's a valuable research tool to give